In this short presentation, we'll explain how we intend to employ the myeloid cell therapy platform technology in the setting of Alzheimer's disease, a neurodegenerative condition leading to cognitive decline. In order to understand how the cell therapy will work in this setting, we need to first appreciate what happens in the normal brain during the aging process. So the normal central nervous system will be comprised of neurons, which themselves are comprised of a cell body and a long axon, and these will deliver electrical impulses. The neurons will be cohabiting with cells like microglia, otherwise known as the macrophages of the brain, and these have a normal interaction whereby the microglia will be involved in the homeostatic control of the environment. There will be budding off of parts of the neuron, which the microglia will take care of in a normal process. These changes in themselves are not usually pathogenic, and in the brains of healthy people in old age, then you will find both accumulation of amyloid protein and phosphorylated tau. So a small amount of these processes is quite normal. However, in the pathological conditions such as Alzheimer's disease, then the picture is somewhat different. So in a pathological setting, you have the same type of processes occurring, but they will occur a lot more. There will be a lot more stress induced by inflammation. This will cause a microglial activation, and this will be accompanied by much larger deposition of the amyloid protein, building of phosphorylated tau fibrillary networks, and axonal leakage from the amyloid depositions, which will then lead to the Alzheimer's plaque formation. These changes will then result in loss of function of the nerve, and this will lead then to the cognitive dysfunction in the individual affected. On this slide you see a representation of what could be expected during the later stage in the pathological process. On the left you see that the cell body of the neuron is now full of phosphorylated tau in extensive neurofibrillar tangles. There will be large aggregated plaques of amyloid protein, at this stage termed senile plaques, and of course there will be a great loss of the nervous function leading to cognitive decline. You'll also see an extensive pro-inflammation, activated microglia and astrocytes. So the idea of this presentation up until now has been to just set the scene, to understand how Alzheimer's pathology occurs, our current understanding of it, and now let's look to see what we can do about it by addressing our research project. So we believe that one potential mode of therapy for this condition will be to transfer pre-activated immune cells with specific activation status into the CNS of an Alzheimer brain, and this will actually stop or ameliorate the process. We believe that an anti-inflammatory myeloid cell phenotype will be the activation state of choice, because we believe that Alzheimer's disease in essence is a pro-inflammatory neurological condition with phosphorylated tau mediated neuronal loss as the key feature. In this picture you see a cartoon of how we believe our therapy might work, where we'll inject anti-inflammatory macrophages or microglial monocytes, and if they come to the target area, then these will ameliorate all the symptoms of the pathology, such as reduce the amyloid burden by phagocytosing the amyloid protein, prevent further tau phosphorylation and build-up of neurofibrillar tangles, and also deactivate the activated microglia. And whether all or any of these processes will be affected remains to be seen. So that's a 5-minute summary of this research project. We hope you find it as interesting as we do to conduct it, and we hope that you'll follow our progress by checking in on this website in the future.